So, good morning, students. Good afternoon, students.
Good afternoon, sir.
So, students, by the time others are joining, do you have any question related to antibodies? Okay, so I hope whatever we have learned till this point, it's clear to you, right? And if you remember, yesterday we talked about the variable region, right? So the antibodies, so by this time you know the antibodies, the first 110 amino acids of a chain, they are the variable region, right? And rest of the structure, it constitute the constant region domains, right? And yesterday we also saw the different types of antibodies, and we also saw some of the differences, right? You remember some had three constant region domains, some had four constant region domains, right? So today what we are going to do is, so you know the overall structure, you know the different types. So today we will talk first about the hinge region, right? Because we haven't yet talked about the hinge region. So we'll talk about hinge region, and after that we will talk about the different types, right? So like there is IgG, but what is IgG doing, how it looks like, right? What is the role, how much of IgG is present in the serum. So there are a lot of things that we need to understand about each of the immunoglobulin type, okay? But before we move on to that, again, like every time we do, we'll have a small quiz, and this quiz is mostly related to the variable region domain, right? So we yesterday understood this variable region domain, right? So after sequencing, what happened, so let me just assign it to you, and students just do it within minutes, right? You won't be needing much time for this. So I have assigned it to the class. So is it visible at your end? I think yes, it should be visible now.
Yes, sir.
So students, just do it quickly. So students, do it quickly, but do it carefully. Okay? Try to understand the question carefully and then give the answer. Okay? हेलो, हो गया? नहीं, मैं ना अभी तो क्लास में हूँ. Students, do it carefully. So students, what I can see is that you are not doing it carefully. Read the questions carefully and then give the answer. And students, start submitting now. So students, start submitting now. Just submit whatever you know by this time. I have got 15 responses. Do it fast. So students, I am just waiting for 1 more minute. Submit your responses. Okay, so let's discuss the answers. Okay, students, the first question was how many CDRs are present in the heavy chain of an antibody molecule, right? So they are talking about uh, in one molecule of antibody, right? So how many CDRs should be there? So each variable region have 3 CDRs, right? So it becomes how many? 3 plus 3, right? So it's 6 because we are just talking about the heavy chain. Okay, so it is 6, not 12, right? So few students have mentioned as 12. So as I said, you have to be very careful. We are not talking about the whole of the antibody molecule, we are just talking about the heavy chain, right? So the correct answer is 6. Question 2, and just imagine only 30% of the class has given the correct answer. So most of you have got confused in this question. How many framework regions are present? Okay, so first thing is we are talking about framework, right? And one variable region has four framework regions, right? So how many? So one would have four, uh, so one would have 16, right? And we are talking about the dimer, right? So you have to multiply it with 2. So the correct answer is 32. Okay? So only 6 students have given the right answer in question 2. Okay?
Sir, excuse me.
Yes.
Sir, at my end, it's showing correct answer is none of the above.
No, it is at my end, it is uh, 32. So I don't know what's the problem at your end, but I think Baki. So what others are seeing?
Sir, 32.
Sir, 32.
Yeah. Okay. Okay, so Devanshi, the correct answer is 32. Okay. Okay. Question 3 is length of constant region domain of IgG is. Okay. So we are just talking about the constant region domain. So the correct answer is 330 amino acid because IgG have three constant region domain and one domain is for around 110 amino acids, right? So the length of constant region domain of IgG is 330 amino acids. So there are three classes of antibody which have a constant region domains of 330 amino acids. IgE and IgM have 440 amino acids. Okay. Question 4. This is the question which all of you have given the right answer. Which antibody can be seen in a pentameric form? So it's only IgM which can be seen in a pentameric form. Rest others cannot form these multimeric forms or the pentameric form. Okay, only IgG can be found in dimer mostly, and sometimes rarely as a trimer or a tetramer. Okay. Question 5 was myeloma protein is. So again, many students have confused here, but the correct answer is myeloma protein is antibody, right? Which is produced from the cancerous plasma cells. So it's antibody. You cannot differentiate between a normal antibody or a antibody which is produced from a myeloma cell, okay? But just because its origin is from the myeloma cell, it is called the myeloma protein. So correct answer is antibody, okay? So I think there are still a lot of things which you need to brush up, okay? You have to be very careful because see when I'm putting in the question, we are talking about a dimer, then you have to think in the terms of a dimer, okay? If I say three molecules, then you have to think in that way, okay? Okay, so overall, not very good score today, okay? But I'm sure you'll be more careful in the whenever I give you such questions in future, okay? Okay, so now let me share the slides with you and we are going to talk about the hinge region. So students, anyone, what is hinge region? Anyone? What is hinge region? Okay, where is the hinge region? Okay, again I see one hand from Manaswini. Okay, Manaswini. So Just switch on your camera and just tell us what do you know about this hinge region.
So am I visible?
Yes.
So the hinge region is basically a region that's usually present after CH1 in some of the antibodies, and it provides elasticity to the antibody. So it's rich in proline and cysteine residues.
Okay, good. So what do you mean by elasticity here?
So are you asking me?
Yeah, you said no, it
Oh,
provides elasticity. So what does that mean?
so mostly that it can allow it to slightly not change shape, but maybe modifies shape a bit. So
Okay.
it makes the body more flexible.
Okay, yes. So flexibility is more appropriate term, and I'll show you how it works. Okay. So let me share the slides. The network today is bit slow. Okay, so is the slide visible now?
Yes, so it's visible.
Okay. So students, I hope you remember yesterday we saw this image, right? So we now know that this is the variable region domain, then you have the constant region domain, and this again we have talked the framework region. So you can see there is 1, 2, 3, and 4, four framework regions, right? And three hypervariable region in the variable region domain. Okay, so I hope this is clear. Okay, now today we start with the hinge region, which is here. Okay, okay. Another thing, not all antibodies have the hinge region, right? So so as Manaswini said, that hinge region is after CH1 domain. Okay, so after the CH1 domain, we have the hinge region. Okay, and this hinge region is only present in IgA, IgD, and IgG, right? And this hinge region is not there in IgE and IgM. Okay, so it is not in IgE and IgM. And as you know, IgE and IgM, they have extra CH domain. Okay, so they don't have the hinge region. Hinge region is only present in IgA, IgD, and IgG, and therefore they have the constant region domain length is 330 amino acids in this case. Okay, because this is one domain, two domain, and three domain, right? So 330, and in this case it would be 1, 2, 3, and 4, so 440 amino acids. Okay, uh, when the sequencing was carried out of the hinge region, it was observed that there is no homology between amino acid sequence of the hinge region and the heavy chain, right? So the hinge region had a very different amino acid sequence as compared to rest of the sequence, okay? And this hinge region, it comprised of many cysteine and proline residues. Okay, so when the sequencing of the hinge region was carried out, so it was observed that in this small region there are a lot of cysteine and proline residues. Okay, and if you remember the pictures we have seen, so there is disulfide bonding at hinge region, right? So it is because of cysteine, right? So cysteine helps in the formation of interchain disulfide bonds, right? So therefore, it's the hinge region at which these disulfide bonds are formed, and it is mainly because of cysteine. Okay, so the purpose of having cysteine there is that it helps in the formation of interchain disulfide bonds, and then it is also rich in proline, which again have a very important function. Proline prevents folding in a globular structure. Okay, so antibody has a specific structure, and that is mainly because of proline. So if you remove proline from it, right, it will get folded into a globular structure. Okay, so the shape will change. Okay, so proline allows flexibility between two FAB arms of the Y-shaped antibody. Okay, and okay, so main purpose is that it prevents folding of the antibody into a globular structure. Okay, so for having that Y shape, okay, and for the formation of interchain disulfide bond, cysteine and proline are there, right? So these are some function, right? But the most important function is that it provides flexibility to the arms of the antibody. Okay, so this is one picture, and I'm sure from this picture you cannot identify anything, right? But I will show you what's there in this picture. Okay, now if you see this, there is a triangle here, right? Kind of. So it's basically antibodies held together, okay, and you can see the angle here is around 60 degree, right? If you see here, so here the angle between the FAB arms is almost 90 degree, right? You see here the angle between the arms, AB arms, is 0 degree, right? So basically this hinge region, it is providing the flexibility to the arms or the FAB region, okay, so that it can hold on to the antigen, right? So depending on the antigen, it may have to open up or it may have to close, okay. So this flexibility is provided at the hinge region, okay, clear? Students, clear?
Yes.
If you have any questions at this point, you can ask. Any questions? Okay. So now we'll start with antibody classes. Okay. So by this time, you know, there are different classes. There are 5 different classes. So now we will talk about each class one by one. Okay. So first one is immunoglobulin G, IgG. Okay. Now it's the most abundant immunoglobulin in our system. Okay, and just imagine 80% of the total serum immunoglobulin is IgG, right? So there are 5 classes, and out of 5, 80% is IgG. So you can just imagine that IgG is present in such large numbers in our body, right? And we also know that there are 4 human immunoglobulin G, distinguished by differences in the gamma chain sequence, right? So obviously the gamma chain or the heavy chain, it determines the class of the antibody, right? So when that region was sequenced again and again, and always 4 different types were observed, right? So they were termed as IgG1, 2, 3, and 4, okay, and they are numbered according to decreasing serum concentration. Okay, so maximum would be IgG1 followed by IgG2, 3 and 4. Okay, now what is the major difference between these four types? The difference is structural. Okay, so in this there is variation in the size of hinge region, and there is variation in the number and position of interchain disulfide bond between heavy chains, right? I'll just show you the picture in the next slides, but just focus on the structural differences. Two major differences. One is the size of the hinge region, right? So in the next picture you will see that the size of the hinge region varies, okay, and then you will see the number and position of interchain disulfide bonds. So the so that is the difference, and you can see here, right? I said two things: size of hinge region, okay, so you can see here the size of IgG3 is big, right? So the size of hinge region, there is variation, and the number and position of disulfide bond. So you can see uh, that there is variation in the number and position of the disulfide bonds in different subclasses of IgG, right? Clear? Okay, so this is the major differences between the four subclasses of human immunoglobulin G. Okay, now what is the role of IgG? Right, we need to understand its biological activity. Okay, now very important role. I think we have also discussed this earlier, that some of these immunoglobulins can cross placenta, okay, and they can provide protection to the fetus, okay, and sometime it can also create problem for the fetus, as it happens in case of erythroblastosis fetalis, but it is basically for the protection, right? So these IgG1, 3 and 4, they can cross placenta and provide protection to the fetus, okay. Then you have IgG3, its most effective complement activator, okay. So once we finish with antibodies, we will start with the complement system, okay. So complement system is, so there are different pathways, right, and that leads to killing of the pathogen, okay. So IgG3, it plays a very important role. So there are two antibodies, mainly IgM and IgG3, which leads to activation of the complement system, okay. Because see, मैंने पहले भी बोला था, what is the role of antibodies? One of the role is binding, okay, but binding of this antibody to the antigen, it will not lead to killing of this pathogen, okay, but the ultimate aim of the immune system is to kill this pathogen and remove it from the system, okay. So what happens in complement system? The antibody, it binds to the antigen. This whole complex of antigen and antibody, it leads to activation of the complement system, and that leads to killing of this pathogen, okay. So we will understand this concept when we study complement system in detail, okay. But just remember that IgG3 is one of the most effective complement activator, okay. And IgG1 and IgG3, they have high affinity for Fc receptors on phagocytic cells, which is good for opsonization. Okay, so again, this is very important. Let me try to explain this. Okay, now students, there are phagocytic cells like macrophages, right? So suppose this is your macrophage. Okay. Now, what happens? These cells, these macrophages, they have receptors, right? Receptors for antibodies. So, let me add few receptors. Okay. So there are many such receptors. So these receptors are, so I think I have already told you about this. When we say FCR, it means receptor for the FC region. Okay. And it could be specific. So if we are putting it for gamma, then we put it as gamma. So it becomes FC gamma R. Okay. So this is the receptor for FC region of gamma IgG. Okay. So now focus here. This is your uh, cell, phagocytic cell. Okay. Now in your system, some antigen came. Okay. Now what happens? What binds to this antigen? So antibody binds to it. Okay. So one thing is binding, right? But as I said, binding alone will not kill the pathogen, right? And it will not activate the immune system further. So what happens? Now what happens? So these are the receptors, right? So what happens now? These are the receptor for FC region. So the antibody binds to the receptor through this FC region. Okay. Okay, so now this complex, the immune complex of antigen and antibody, it moves towards the phagocytic cell, and this complex binds to the phagocytic cell through the FC region, right? So the receptor is binding to the FC region of the antibody, and that's why these receptors are called as FCR. Clear? And if this receptor are specific for a specific type, then we put the name of the chain, right? So it could be epsilon or it could be delta, right? So now what happens once this binding occurs? Okay, once this binding occur, this whole complex is taken inside the phagocytic cell in a vesicle. Okay, so and then here this pathogen is killed and broken down into smaller pieces, and this obviously we are going to discuss in detail when we talk about antigen processing and presentation. Okay, but I hope this concept is clear now.
Sir, can you please repeat?
So what is the point that is not clear?
Sir, the FCR thing.
Okay. Okay, so FC means the FC region, right? And when we put R, so it's the receptor. So we are now talking about the receptor of FC region of what? So suppose uh, we are talking about the receptor for IgA, so it would be alpha here. So FC region of IgA and receptor. So where this receptor would be? This receptor would be on a phagocytic cell, right? So suppose you have this phagocytic cell. The phagocytic cell will have a receptor, right? To this receptor, what will bind? Antibody will bind. Okay, so let me change the color of antibody. So now you have an antibody, right? Where this antibody will bind on this phagocytic cell, it will bind on the receptor, right? Now just imagine this FAB region is already attached with the pathogen, right? So which end is free? The FC region, right? So this whole complex will bind to the phagocytic cells through the FC region, right? So the FC region has to come into contact with the receptor. Okay, clear?
Yes, sir.
And these receptors are called the FC receptors. Okay?
Yes, sir.
And these receptors and the binding of the antibody to these receptors, that lead to phagocytosis of the pathogen. Okay, and these phagocytic cells like the neutrophils, dendritic cells, macrophages, they are good in killing the pathogen. Okay, and how they kill we will discuss in a separate class when we talk separately about these cells. Okay, so just again just giving you an overview. So there are four types: IgG 1, 2, 3 and 4, right? And it was mentioned it has been numbered according to the concentration, right? So in our body 66% immunoglobulin G is IgG 1, okay? IgG2 is 23%, IgG3 is 7%, IgG4 is 4%. Okay, which of them can cross placenta? So you can see IgG1, 3 and 4 can cross placenta, but IgG2 cannot. Okay, complement activation. So I just told you that IgG3 is the best activator. Okay, followed by IgG1 and IgG2, but IgG4 it cannot activate the complement system. Okay, and with respect to its binding affinity on with the FC receptors, so IgG1 and IgG3 I have high affinity. Okay, so I think you just need to remember this much about IgG. Okay, so any questions? Okay, let me have your attendance, and in the meantime if you have any question you can always discuss. So Adish,
Present, sir.
Atoiba,
Present, sir.
Ayushi,
Present, sir.
Sam,
Present, sir. Present, sir. Am I audible?
yes, Devanshi,
Present, sir.
Ishita, Ishita, okay, Janvi,
Present, sir.
Kshitij,
Present, sir.
okay, Ishita,
Okay,
okay,
sir.
Ishita, yeah, Manaswini,
Present, sir.
Namrita,
Present, sir.
Nandini,
Present, sir.
Nehal,
Present, sir.
Rupal, Sahiba,
Present, sir.
Sanjana, Sanjana, Shakti,
Present, sir.
Shivani,
Present, sir.
Tanvi,
Present, sir.
Tridev,
Present, sir.
Vivek, Vivek, Yukta,
Present, sir.
Yuvraj.
Present, sir.
Okay, fine. Okay, students, any questions?
Yes, sir.
Yes, what's your question, Shakti?
Sir, what about the flexibility of IgE and IgM since they lack the hinge region?
Okay, so even they are flexible, but obviously the flexibility is less. Okay, and just because they doesn't have that hinge region, it doesn't mean that they don't have cysteine and proline. Okay, it is there, but you cannot distinguish them as a hinge region, right? And yes, they also have flexibility, but not to that extent what you see in other three types. Okay?
Yes, sir. Thank you, sir.
Okay, students, so I think we can end the class now. So, see you in the next class, okay? So I'm stopping the recording of the class.
Okay, sir.